Okay, so one more thing. This school is being organized by the ICTP-SAIFR here in São Paulo and the ICTP in Italy. So it's a joint event. So it's very nice to have that. And I think that's it. Anything else I should say? No. So okay. So we start with Luca Peliti. So thank you again for coming. It's a pleasure to have you here. I'm going to set up with the microphone. You put this on the foot. Go with it.
Is it working?
Yeah.
Okay. So the fact that there is a 30 minutes discussion after the 1 hour lecture doesn't mean that you are not allowed to ask questions during the 1 hour. The only thing is that because I have a problem with ears at the moment, your question loudly. Otherwise, I don't hear, don't understand what you're doing. So what I'm going to present to you is just an introduction, mathematical methods of population genetics and evolution. From, say, it will be sort of a very, I hope, a very easy, fundamental introduction that is a setup for more advanced subject that we'll see later. So let's start from one thing, which is, say, there's an ongoing experiment which was started by Lenski now about 30 years ago, of following the evolution of 12 populations of E. coli bacteria over and over again with, with the cycles of 24-hour cycles, more or less, except for weekends, looking in detail on how this population changes in time in an environment which was originally quite a difficult environment to adapt. So the idea is that you can have a very detailed information about the genetics of this population, and you can follow this in time, so you can have, say, the uniqueness of evolutionary history is amenable to careful experimental analysis. And you have the luxury of making a prediction, and then you can test it. It's almost like physics. So the idea is that you start from an ancestral population of E. coli, lab strains of E. coli, and then these strains are divided into two substrains that are differentiated by say, a way of being able to understand which of the two subpopulations they belong to. Let's not get into the details. And each of these subpopulations are then divided into 6 subpopulations each, so that you have 12 populations evolving in parallel, and you can distinguish which is which, say, macroscopically. And the idea is of having these two subpopulations that you can be distinguished macroscopically without going into the genetic details is a way of checking about say, the contaminations of different populations together. So what you do is you start, say, you evolve this population by putting a small amount of, say, 0.1 milliliter of the previous solution into 1 becquerel with 10 milliliters of nutrients, actually very poor nutrient solution, wait 24 hours, then you take again 0.1 milliliter, you put it in a different beaker with a fresh solution, and then you go on and on and on every 24 hours for 30 years or so. And then you look what happens. So this is a parallel propagation of 12 independent populations, and with the standard density of, say, 50 million cells per milliliter. End up, this is old slide, so it's about 10 years old. So already after about 20 years of evolution, you had 12 independently evolved population in glucose-poor environment. So this is why the environment was challenging for these poor bacteria. At that time, it was 40,000 generations, and a number of, say, billions of simple mutations have occurred for each strains in each subpopulation, probably several times. And at that time, only about 10 to 20 mutations reached fixation during the experiments. Some strains had evolved into hypermutations. They have a much higher mutation rate than the wild type. And one point is that the one major innovation, that is the ability of metabolized citrate, which is usually not metabolized by E. coli, has developed around, say, after 31,500 generation. You can see it in this. So here you see that this beaker has, say, sort of higher turbidity because it has evolved to use the citrate which is present in the media, whereas the other ones are not able to use that. And this is a very major innovation for E. coli, because this is one, say, one of the traits that distinguish E. coli from Salmonella, for instance. So this, what you can do is now you have, this is just at the beginning, in fact, of a series of experiments that have been done in which you can go in the details of the evolutionary mechanism and check what happens both at the phenotypic level and the genotypic. Now what we are going to do is just to consider the basic mechanism of evolution, see how we can describe them mathematically and model and make model of them. So reproduction is, of course, the basic mechanism of living system. And if you introduce, so now we are going to describe some details of reproduction for the special case of bacteria. You introduce bacteria in a fresh solution. There is the first time a lag in the time in which apparently nothing happens, and the number of bacteria in the solution does not change with time. This lasts, say, can last up to a few hours, and then at a certain point it starts, the number of cells starts increasing exponentially until it reaches some level, say it's called the carrying capacity, and that there it remains stationary for a long time. You see, of the order of 1 day or so. At that point, is the you start having the cells start to die, and then they die off pretty fast on the time of several hours. You see that the stationary phase is reached after about, say, 12 hours to 15 hours. So that means the protocol that I have described before, in fact, what you end up is that the cells, the population had been for several hours in the stationary phase when it is inoculated in the next beaker for the next 24 hours cycle. So let's first look at the exponential phase. In this exponential growth phase, the generation time is about 20 minutes. So every 20 minutes you can, and so let's make a very naive estimation of what is going on. The expected population at a certain time t will be given by the original population size times 2 to the power integer part of t divided by this tau generation time. This assumes, in fact, that the cells are so they, like a battalion, they decided to reproduce all together. Of course, this is not what happens in fact, but let's start with this very naive idea. Now, assume that the cells reproduce on average once within this generation time, and then what you have is that the number of individuals, so if you forget about synchronization, you get something of the order in which the number of individuals time t will be the integer part of n0 times this number here, and there you have it's more continuously increasing. And in fact, I say this is just to show you that we are going to change from a representation which we assume that there is a synchronization non-overlapping generations to a description in which you have a continuous time evolution. And we're going to play with that because sometimes it's easier to make the mathematics in one case and sometimes make a mathematics in the other case. Let's go again, look at this, and let's assume that this is what really happens over and over again. So if we take a 20 minutes as a generation time, we have 72 generations in one day, and then from one individual in 2 days, we get a 2 to the 144 individuals, so that makes a 2 to the times 10 to the 43 individuals, that makes say 10 to the 28 kilograms, so that's 3,000 times the mass of the Earth. So that means that is why the exponential increase can only last for a limited amount of time. We didn't, in this description, we imagine that as the bacteria reproduce, they don't die. So the first thing we want to understand is what happens if there is a non-vanishing probability for the cells to die at each generation before reproducing. So let's assume that now, say, reproduction is not like a battalion, is not, say, deterministic, but it is a random process, and also death is a random process, and we want to know what should we expect for the evolution of the population. And this is called the Galton-Watson process. So let's just first start with discrete generations. So in which the reproduction and death happen in synchrony. Let's ask what is so we defined the probability Pn, that is the probability, you start with a single cell and you ask what is the probability that this cell has exactly n offspring in the next generation. This is something that is defined, say, by the metabolism of the cell itself. So it is a given, say, in a given environment for a given kind of bacteria, and assuming that there are no interactions, which is what we are assuming at the moment. So of course we have the P0 is the probability of death, and the P2 is the probability of reproduction, and then you have a normalization. Then you ask, what is the probability that you start from a single cell, you wait T generations, and then you ask that what the total population size is equal to N after T generation, assuming you start from a single cell. This is one quantity you want to evaluate, and the other quantity you want to evaluate, which is related to that, is the ultimate extinction probability. What is the probability? You start with one cell, and then eventually you find no cells at all, the offspring has died. How would you evaluate that? Well, this is you consider the process after T generation with a given population size, say small n here. Now, we start with n equals 1 at t equals 0. Then at t equals 1, you have n equals k with a probability pk, which is that probability that we have defined before for the single generation step. Then the probability that the issue of one of the k individuals who taken as independent process, so this is the assumption that we are making in this moment, is given by, so the probability that the issue of the one k is equal to m is given by this pm of t minus 1, where pm is the quantity that we want to evaluate, all right? But it's evaluated t minus 1 because we have moved one generation ahead before. And therefore, we have this kind of equation which relates a pm of t, probability that the size of the population is equal to m, is in terms of a pmj of t minus 1. And what is this? It's the probability that at the first generation you have k offspring. Then for each of these k offspring you ask what is the probability that you have m1, m2 to mk offspring after t minus 1 generations. And this is the probability of having this distribution of offspring for each of the descendants of these k individuals of the first generation. You condition on the fact that the total population is equal to n, sorry, should be an n, and you sum. And we want, so this is an equation that we want to solve, and it is, you can solve by going to a generating function. So you define this quantity that is simply the sum over population size, z to the n of this probability you want to evaluate, and the analogous quantity for the number of offspring in one generation. And then you find that this very nice equation that gamma, generating function of the generation t, is obtained by applying this function, that is the function, generating function for a single generation of offspring, applied to the same generation function, but a generation t minus 1. And therefore, by iterating this mapping from gamma via g to a different gamma, we obtain the solution of the problem that we have. The generating function at the generation t is obtained by applying this function g t times to the initial value. And the initial value with only a single bacterium is equal to z itself, where z is the parameter that we have used to define the generating function. And of course, the ultimate extinction probability is obtained as a limit of this generating function when z is equal to 0. And we can now look at what is the solution. We have, in this case, we have z is the parameter of the generating function here. And the g of z is that the generating function for the number of offspring in one generation. And we have, you can show that this is a convex function, and it must be equal to 1 when z is equal to 1. In one case, like this one, there is only one intersection between g of z and 1 that it corresponds to 1 here. And in this other case, we have instead that we have one intersection here, but there is one intersection z star. And now imagine that you start from a given value of z, whatever, and then you iterate. If you iterate, for this one, you find that this goes up, 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 up again, and goes to 1. So if the average number of offspring at any given generation for a single bacterium is less than 1, then you find that the limit of the generating function is always equal to 1, and therefore that the extinction probability is equal to 1 itself. So probability 1, your population goes eventually extinct. On the other hand, if the average value of the number of offspring is larger than 1, then the limit of this generating function is equal to z star, And this is also the probability that the population becomes extinct. We can play it also in continuous time, but I'm not going to do that. And it's you can do that as an exercise if you like. But what is more interesting is what happens if you ask a question. You are given a population of a given size. What is the probability that this population does not get extinct? So in this case, we are not starting with a single bacteria, but with n bacteria. Assume the simple thing, that there is only a death probability, pi0, and the probability of reproduction, pi2, and discrete generation. Define this quantity S is 1 minus the ratio of death probability divided reproduction probability. And this survival probability is the eventual survival probability, that is 1 minus the extinction probability, is called Sn. Then what you have is that Sn is given by, you can evaluate it. There is a probability that some individual dies, and so in this case, the size of the population goes to from n to n minus 1. And there is a probability that one of the individuals reproduces, and therefore you go from n to n plus 1. And you can use this definition and find that this is an expression of the corresponding probabilities that you have. And of course, the probability of eventual survival is given by the sum of the probability of eventual survival when the first step, say, is 1 death, plus the probability of eventual survival when the first step is reproduction. So you get this equation here. With the boundary condition, of course, that S0 must be equal to 0 if you start with no individuals, and limit as n going to infinity of Sn is equal to 1. And therefore, for whenever S, this quantity here, is larger than 0, you can solve this equation easily, and you find that Sn is given by this expression here. And whereas when S is smaller than 0, this is a quantity that should say becomes negative, but in fact the only thing that makes sense is it is equal to 0. And that corresponds to the case in which the average value of offspring is less than 1. And in particular, if S is larger than 0, you find that starting with one single individual, then this survival probability is equal to this quantity S that I have defined before. And then you see that this is a very simple result, and it is nice to keep it in mind because we can use it in different contexts and in different and sometimes surprising contexts. The other question that we can ask is that, okay, we are given a certain, we would like to know how many individuals should be there of a given population to be almost sure that the survival probability, say that the population eventually survives. So say, what is the minimal population size n star such that the survival probability exceeds 1 minus gamma? Let's say that the extinction probability for n equals 1 is this dex z star that we have evaluated from the Galton-Watson process. And this is something that we can evaluate from that graphical construction that I showed you before. What is the probability that all the n lineages from this starting population go extinct? And this is given to z star to the power n. But these quantities must be smaller than, say, 1 minus gamma, where gamma is essentially arbitrary. So n must be larger than n star given by log gamma, divided by log z star, because gamma is the extinction probability. You want to have it as smaller as possible. And so in particular, when only the death and reproduction probability do not vanish, we have that the Z star is given by this expression is equal to 1 minus S, which is again the same quantity that we have defined in the previous slide. And therefore, you put this and you get expression N star is essentially of the order of log, absolute value of log gamma, where gamma is this extinction probability we want to make as small as possible, divided by S. And this is again a result that we are going to use. So the nice thing is that it depends very strongly on this S factor and much more weakly on the gamma that is the threshold that we have put to the extinction probability. Of course, the exponential, all this was done in the exponential phase, assuming that there is no limitation to the possible growth of the population size. So this is, of course, not going to, as we have seen before, is not going to last for long. So what happens if we take into account the fact that the resources are limited and the population cannot exceed some value? This is a classic equation called, so this case has been described by the Malthus-Verhulst equation. And in this case, we're using a continuous time setup, and we assume, for simplicity, we take n to assume real values so that we can use the differential calculus. And so the simplest case that we can make is that the reproduction rate decreases linearly with n. Of course, we can also make a different and not completely equivalent assumption, say that the death rate increases with n. So, but let's take this case in which a reproduction rate decreases linearly with n, and we forget about fluctuations. Then we can write down an equation for the population size that is in this way. We have some sort of reproduction rate here times n, but this reproduction rate is multiplied by 1 minus n over a quantity, positive quantity k, that is usually called the carrying capacity. And you can solve this equation easily, and you find this expression here. And this expression has the well-known logistic form. So it starts exponential, as we have seen, with the rate r, and then it has an inflection point and eventually reaches a plateau which corresponds to the carrying capacity. And in this case, I just put the numbers that correspond to this long time evolution experiment, that is r is equal to this, say, correspond to a generation time of 20 minutes. We have initial value is 5 million individuals, and the carrying capacity for 10 milliliters is 5 times 10 to the 8. And so this is what you expect from the Malthus-Verhulst equation for the case of this long time evolution experiment. And of course, it does not fit with the data, because if you go and look carefully what happens in the experiment, you find that the stop here is not so, the stop in the actual experiment is not so gentle, it's pretty abrupt and arrives with something that looks much, much more abrupt than this guy. But it's starting point. Now, selection is the fundamental idea of the evolutionary theory. This is because there are all these portraits of Darwin in his later life. I like this one because this was taken very soon after he came back from the Beagle voyage. And I think it is also is nice. But then he had, so this was in the 1840s. So when he was actually developing the theory, and he had a lot of trouble, health trouble, and his daughter died, so it was a very stressful time. But during this stressful time, he developed his theory. So you see, you can see that there was something. And this is the key passage of the big book of the origin of species. And just wanted to mention one thing. Say, individuals having any advantage, however slight, would have the best chance of surviving or procreating their kind, and we may feel sure that any variation in least degree injurious will be rigidly destroyed. And this preservation of favorable variation and rejection of injurious variation I call natural selection. That is the key idea. And then there is a very nice add to it. Variations neither useful nor injurious would not be affected by natural selection and would be left as fluctuating elements, as perhaps we see in the species called polymorphic. So in the same page, Darwin introduces the idea of natural selection and say, well, but it doesn't cover all the cases. You also might have what is called the neutral cases, and you can find it in this species called polymorphic. And this is a commentary by Huxley. My reflection when I first made myself master of the central idea of the origins was how extremely stupid not to have thought of that. So in order to model this process, it is customary to introduce a quantity, say a measurable quantity that determines how a phenotype is, say, favorable or unfavorable to a given life form. And this is called the fitness. And then let's take a very simple case in which you have two kinds of individuals, A and B, or one with population NA, the other one with population NB, discrete generations. And you define a fitness as the expected, so the average value number of offspring of an individual of type I, and where I is A and B. Let's call this WI. So, and then assume, first, all the individuals reproduce independently. Second, the offspring of an individual has the same value of W as its parent. And third, we neglect fluctuations for the time being. Then what you have is that the number of individuals at generation T plus 1 of type I is equal to the number of individuals of type I at generation T multiplied by this fitness value W. Let's look at the change in the overall composition of the population. In this case, we define, so the fraction of individual type I at generation T is given by number of individuals of type I divided by total number of individuals. And then you start evaluating, well, how does this fraction change? Well, you plug in the definition, you use the definition of fitness that I have introduced before. So you see here we have W A times NA, and then you have something downstairs. And then we can, you can sort of introduce a definition of the fraction. Here we focus, for instance, on the X is the fraction of type A. And eventually what you get is this equation, that the fraction of individuals of type A at the generation T plus 1 is given by the fraction of individuals of that type at generation T multiplied by the fitness of type A divided by the average fitness of the population, where the average fitness of the population is given by the weighted average of the fitness times the corresponding fraction of individuals for different types that you are considering. And again, let's write this in a slightly different way. Let's say that the WA divided WB is equal to 1 plus S, assuming that A is the favored variant, then S is positive, and then you have that X at T plus 1 is equal to X at times T times 1 plus S divided 1 plus S times X. X is between 0 and 1, so this quantity is larger than 1, and therefore X increases in time. And this is the basic mechanism by which the favorable type invades the population. There is another quantity we might wish to evaluate is the mean fitness in the population. Let's assume a more general setting in which we have R different types, each of them with their fitness. The mean fitness is given by the weighted average of the fitness of the different types. And then it is a simple exercise to show that the change in the mean fitness is given by the ratio, say, of the variance of the fitness, which is a positive quantity, divided the average value of the fitness, which is also a positive quantity, and therefore this quantity increases unless the variance vanishes, and the variance vanishes when essentially all the individuals you have in the population have the same fitness. And this was very modestly called by Fisher the fundamental theorem of natural selection, and he made this kind of remarks. So he made a more complex statement, but let's say put it this way: the rate of increase of fitness of any organism at any time is equal to its genetic variance in fitness at that time. Professor Eddington has recently remarked that the law that entropy always increases holds the supreme position among the laws of nature. It is not a little instructive that so similar a law should hold the supreme position among the biological sciences. So this is, to put it, a slightly controversial statement. So Carl Sigmund made, so in physics we see disorder, so entropy increases, disorder growing inexorably in systems isolated from their surroundings. In biology, fitness increasing steadily in populations struggling for life. Ascent here and degradation there, almost too good to be true. And so some dissent about the importance of this theorem by Maynard Smith say, my own view is that it cannot play an important role in biology. Okay, so this is what happens in continuous time. The only thing that I want to know is that what happens in continuous time, instead of having this ratio, what you have is that the increase in the fraction of individual type K is given by the, so first of all you have to define what is called Fk, which is a different definition of fitness. Wk is the expected average number of offspring of a given individual in a given situation. Fk is essentially the log of that number of offspring divided by the generation time. So I call and sometimes I use W is a right fitness, W right. So for Sewell right, evolutionary, and this is a Fisher fitness. And the Fisher fitness can be positive or negative, it doesn't. And the evolution of the fraction of individual type K is given by it's the same fraction times its Fisher fitness minus this time the average value of the Fisher fitness. And the fundamental theorem takes this place. The change in time, as a derivative with respect to time of the Fisher fitness is given by the variance of the Fisher fitness itself. And again, this is a non-negative quantity. In the Malthus-Verhulst regime, again, let's look at what happens in the continuous time. We have an equation like that. And what we have here that is quite interesting, you just plug it in and you sum, you find that the rate of increase of the population depends on the composition of the population. It's no more independent on the composition of the population. But in fact, what is more important is that you have, in the case in which you have a carrying capacity, you have a separation of time scales. So pretty fast, you have that the total size of the population reaches a value which is close to this carrying capacity. At that point, the total number of individuals remains pretty much constant, and then it's only the composition which changes over a longer time scale. And this is really the regime in which we are supposed to say, which is more relevant for evolution. I'm afraid I have to jump on this thing and say, how do you measure fitness in these long time evolution experiments? There are a number of things that, let's instead continue to model what is going to happen. So we can have, I define fitness forgetting about details, but sometimes you have a fitness depends on the composition of the population. And this is, there are a number of interesting questions you can ask. Here I'm just going to give you some definitions because they are relevant and important, and we'll see later. So you might have a situation, imagine that you have a population in which there are only two variants, A and B, and you might have a situation in which the fitness of, say, A decreases as the fraction of individuals carrying this feature A increases in the population. So we have, in this case, a negative frequency-dependent selection, in which, for instance, you have, this is the Fisher fitness that goes down as XA increases between 0 and 1. And you see it crosses 0 at some intermediate value. So at this point, you have that there is a fixed point for the composition of the population. The population will have that the fraction of individuals carrying A will stay put at this value of the frequency. And of course, the remainder will be, say, the B kind. In this case, so we see that if we have negative frequency-dependent solution, then we have eventually a coexistence, and the population will be polymorphic, but by a different mechanism than what was mentioned by Darwin. The other possibility is that you have that the Fisher fitness of A increases when it is more and more frequent in the population. Here again we have a crossing here, but now you see that this frequency becomes is negative here and positive there, and that means that if you start from this point here, you either go down to eventual extinction or you go up to what is called fixation, when all the individuals of the population carry this feature. And that means that this XA star here, which is a fixed point of the dynamics, is in fact unstable. And this is called a disruptive selection, and you have a bistability. If you start with too few A individuals, you end up with extinction. If you start with a higher number of individuals, you go to fixation. Now, the setup for the frequency-dependent fitness is usually called evolutionary game theory. And there are a number of games that have been played by, for instance, consider what are the situations in which it is likely that you have this frequency-dependent theory. And one of the most popular one is called the prisoner's dilemma. The prisoner's dilemma is the idea, well, you have two people who have committed a crime, and then they are arrested and taken separately, and then each of them is asked whether they want to denounce his complicit or not. And the idea is that if neither of them denounces the complicit, they will have, say, a weak punishment. But if the accomplice denounces you, then you are going to have a very heavy punishment. And the question is that rationally, each of them will have, and then on the other hand, that it will be set free, so the accomplice will be set free. So the idea is that it is rationally convenient to denounce the accomplice, but in this way, both of them will be worse off. So imagine that you have a situation in which, in terms of fitness, the fitness depends on the encounters between, say, two different strategies, a cooperator and defector. When both are cooperating, then you have some sort of value of fitness as 1. When a cooperator meets a defector, then it gets a value of fitness which is 1 minus S1, but the defector gets 1 plus S2, and if the two of them say the defector meets a defector, then they have 1 minus C, where C is positive, and therefore both of them are worse off. And the point is that if this is the case for this kind of game, say why cooperation is maintained in nature. And the funny thing is that you can have situations in which you might interpret data as being some sort of, say, life forms playing a kind of prisoner dilemma. This is a famous case in which you have a phage. A phage is a virus which infects a bacteria, and a phage which is a high multiplicity of infection. So a situation in which you have several phages, several virions, say, infecting the same bacterium. In this case, it will pay for the virion not to produce some of the relevant proteins that are needed to infect, and instead multiply as fast as possible in order to make many more copies of itself than producing the protein that is useful, in fact, for the keeping the infection. And so in this case, what you have is that depending on the, if you have only few virions, then this is not a very good strategy. But if you have many higher multiplicity of infection, this becomes a very relevant strategy, because the copies of the ones who are, say, copies of the defector will become more frequent. And so in this case, what you find is that, in fact, by you can evaluate the mean fitness as a function of the initial frequency, and you find that the mean frequency, the mean fitness of the virions decreases as you increase the fitness of the different two types. So this is a situation in which you find, say, that you have that, in fact, apparently there is a decrease on the overall mean fitness when the, say, defector are initially more frequent. There are other cases that are well known of frequency-dependent selection. This is a nice work on. So here we have some lizards in California. They have different strategies. I don't want to enter in what strategies. Say it's a sort of mate competition strategies. But the funny thing is that these the strategies are associated with some visible phenotypic traits, colors, say. And they play a sort of game of rock, scissors, paper. So one strategy beats one of the other two, but is beaten by the third one. And in fact, this Sinervo and Lively over several years in California so you have the orange, so can beat the blue can beat the yellow beats the orange. And these are the data. So these is a representation of the fraction of individuals of different colors, or if you like, different strategies over several years. So it starts in 90, 91, 92, 93, 94, and what you see is a sort of cycling behavior with a drift, and this corresponds, in fact, to the prevalence of successive strategies over and over again, like playing a rock, scissors, paper. And you can put that in a sort of evolutionary model in which the fitness, fitness depends on the composition of the population, and you'll see this kind of oscillations. They eventually, over a very, very long time, the model predicts some sort of stable equilibrium, but of course at that point the model itself is probably unlikely to be realistic. So what happens to the fundamental theorem if you take into account the frequency-dependent selection? And well, you can write it in this way. And so So you can, so what you have is that here you have the term that we had before corresponding to the variance, but here we have an extra term which in fact can change, and that means that there is no need, there is no how to say, in the presence of frequency-dependent selection, the fundamental theorem has to be qualified in the sense that you don't have necessarily an increase of the mean fitness. And I think that is more or less what I wanted to say for today.
Could you go back to that question?
Huh?
Could you go back to the so the point is that this extra term does not have a sign.
You don't have, not only you don't have the fitness itself is not always increasing, but in fact you can prove that there is no Lyapunov function.
There's no what?
Lyapunov function in general. So there is no reason for being, for reaching a single point, a steady state. Okay, and I think that I'll stop here. Okay, thank you.
We have time for a few questions before the break, if you have some comment or...
Just a little thing in this business dilemma.
Yeah.
They should be symmetric.
No, it is. In this case, okay, the prisoner dilemma, as I showed, is symmetric because it is really between justice.
Because you had S1 and S2. One is 1 minus S2.
Yeah,
the other one
it's say
1
the
plus S2.
Yeah, but
1 minus 1.
Yeah,
so it's not symmetric.
It's not symmetric. Okay. Yeah, because
I thought it was symmetric.
No, but it just depends. Say the strategies, the payoff matrix that depend on which strategy you use. Yeah. Indeed.
There's also this horizontal. Genes can go horizontal.
Horizontal gene transfer, but I didn't say anything about genetics here.
Yes, yeah.
Yeah, so this is. I'm going to discuss that a different time.
To come back to prisoners.
How is fitness evaluated at rock, paper, scissors dynamics?
Sorry, I don't hear.
How is fitness evaluated at rock, paper, scissors dynamics? Like, how can you say that some...
In this case, there is no quantitative evaluation because the data are in fact field data. What you have is that this behavior corresponds to some sort of mating control behavior, and you can argue that one beats the other, but there is no quantitative data for that, for that specific case. I mean,
But if you would model it, like how would you compare fitness in something in populations that one is more advantageous than the other, but there are the...
Well,
there is the
say what you do to evaluate fitness in the wild is very hard. What you have to do is to look at the actual change in the population, which is actually your sampling population, so with enormous details, and try to fit it to some sort of mathematical model. That's the only way you can do it. And this is what they did. I mean, this is why... In the case of the long-time evolution experiment, what you do is you inoculate a plate in some condition at the same time with some sample from your actual population, some sample from the frozen ancestral population, so that you are able to evaluate the number of individuals, say the growth rate. Well, not exactly, but say the growth rate ratio between the actual population you see and the ancestral population, because you need to have some sort of unit measure. Absolute values of fitness don't make sense, basically.
Maybe his question has to do with what the payoff matrix looks like for this game. I don't know, but we can...
Yeah, but you say you have to guess it,
Sure,
basically.
yeah,
There is no way of measuring it. In this case, there is really no way, because it depends on the behavioral trait. So it's something that you can do.
So should we break for 10 minutes? Yeah, okay, so we have a 10 minutes break and then we come back for more questions. Okay, thank you. Okay, so this is the time where we should ask questions, things that you haven't understood well, or comments, anything. And if you don't have any questions, I'm going to get one of you to come forward and explain all the math here.
Yeah.
Thank you, Luca, for your presentation. So, going back to that separation of time scales, right? You said that first, the population reached fast the carrying capacity, and later you had that the population tended to the fittest, right? So, has that been compared to what happens in nature? Like, do we have biological support for that?
The separational time scale is there, say all the time, yes. The details depend on the model. As I mentioned, you should not take the Malthus-Verhulst because the population dynamics does not fit data. But really what you have is, by and large, the natural populations have carrying capacity. They are close to their sizes are close to the carrying capacity, and most of the change happens in the composition over a longer time scale. Of course, there are exceptions. I mean, one exception is humans that change carrying capacity suddenly in a number of times, and okay, you shouldn't, but
so what do you think happens there once the population is fit? You know, once every individual is fit, how will
there is sort of a fluctuation in census fluctuations going on, but that is over orders of magnitude much smaller than what you have. You have this exponential increasing phase. So the exponential increase you only have when the life form, say, gets into a completely virgin space. There is some sort of bias. People are fascinated by the exponential increase phase, but this is really not what happens in the wild. In the wild, what you have is that populations stay close to their carrying capacity, and then there are a number of fluctuations to the composition, but not so much on the side.
So when you talked about frequency-dependent selection, when you have positive frequency-dependent selection, you mentioned that it could lead to several possible equilibria, including disruptive selection. And what about directional selection?
About?
Directional selection.
Well, the idea of selection is the fact that games. There are several mechanisms for that. extreme case of very badly, then they can only survive if there are enough collaborators cooperating. The anticipation that happens in anthology. And I think that microphone is not on?
Oh, maybe if you move it a little bit up.
In colony that there are situations in which a certain, say, minimum amount of some product in order for the whole colony to survive. Below a certain level, then they'll die. I think that Paul these kind of problems.
What are your thoughts on the concept of fitness? Is it a useful currency for dealing with evolutionary change? I mean, you mentioned a moment ago that, exponential dynamics and so on. I'm interested to know what you think.
Well, say that this is really one of the problems. It's very useful for making models and solving differential equations. It's probably not so useful when you're actually trying to understand what is happening. And the reason is that, say, you cannot really hope to collect all the information relative to survival of a life to one single number. And then to compare, I mean, and those things, just comparing them as in the makes much sense. So this is what is called the problem of, how to say, scalar fitness. I don't believe in scalar fitness, let's put it this way. But it's useful as a starting point.
If you could start again, well, you could start again any time.
But, I mean, I'm not asking to start again the lecture, but what would be more useful, do you think? Or you mentioned some measure of the entire performance of the organism, but what might you think about as alternatives to fitness?
I don't have an answer.
Okay.
It's one of the problems. I don't have an answer.
I have a more technical question. Can you go back to the beginning of your lecture when you talked about the generating functions? Could you go through that again? I'm not sure I got that. Yeah.
This one?
Yeah, this one. Can you explain this equation below here again?
This one?
Please. Yeah, that. No.
This,
go back one more. Let's see.
This is in continuous time, so
yeah.
This is okay. This is a fundamental equation.
Yeah, that one.
Okay.
Yeah.
Okay, so the idea is that what you know are these pKs. These pKs here is the probability that in a given generation, one individual has k daughters. K can be 0. All right. Now, this should be a small n. Pn of t is the probability that you start with one individual at generation 0, and at the generation t you have a population of n individuals. Now what you do is that you just start with one individual, you wait one generation, and this individual has k daughters. At that point, you look at what happens after t generations. So you have, instead of having one individual at generation 0, you have a k individuals at generation 1 with probability pk. Now you ask, well, okay, what is the probability that the total population size is equal to n? This is given by the probability that individual 1 at generation 1 has n1 descendants after t minus 1 generations. Individual 2 has M2 descendants and so on, till individual K has MK descendants. And of course, this probability must be, so what you have to do is to sum over all possible numbers of these descendants such that the total is equal to n. So this is this equation. Now, this equation, in fact, can be solved using the generating function. So first you define the generating function for this population size simply by multiplying by factor z to the power n and summing overall. And so what you have is when z is equal to 1, this must be normalized to 1. And then you do the analogous thing for the number of daughters. And then what you take the equation that I wrote before, and you find that if that is true, then summing, here you see, we sum over population size. So summing over population size gets rid of that awkward delta function that imposes the total population size. And what you have is a very simple result, that this becomes sum over k, p of k, probability, times this expression, which is again the generating function for the population size, but evaluated after t minus 1 generations. So what you get is that the generating function at generation t is equal to this function g applied to the generating function times t minus 1. And that is that equation. And we solve it graphically.
Yeah, okay. And that solution that you showed is for the case where you have only P0 and P2. So G
yeah,
is...
yes,
yeah,
but
okay.
of course, I say this is a method that you can use.
Right.
So in this case, and then it is trivial to see that the slope around 1 is equal to the average value of the number of average value of the number of daughters. So here you have a very small slope. You see, if the slope is less than 1, the curve starts above the diagonal, and if the slope is larger than 1, it starts below the diagonal. And this is why you get this extra intersection.
Thank you.
Luca, when you presented your results on this fitness-dependent and for the prisoner dilemma and for the lizards, for me that's population dynamics, and I expect to see cycles and all that. And the fundamental theorem of Fisher is population genetics, where I've never seen cycles or periodic behavior. So I wonder, is there a mixing between population dynamics? For me, the dynamic of those lizards, which we measure in
the composition. I mean, this is in the composition. In fact, you can plug this if you neglect fluctuations, you can plug this in some sort of equation of population dynamics, but within the composition. This is kind of thing that you find. Now, the problem is that most of the time you'll end up with some sort of fixed point, but this is not necessarily the case in general. But I don't know if that is, I didn't hear well, unfortunately. So I don't know if that is the question.
No, okay, you can discuss that later. Because my feeling is that this study of these lizards, it took about 1 decade. You showed the years.
Say quite often you end up with some sort of fixed point.
Really, I mean, for lizards, you can't see any genetic change. You are just seeing the game, I mean, the dynamics between 3 different species. So, like a predator-prey. So that's population dynamics. So why people are mixing these sort of results with the standard population genetics of Fisher, White, and so on? That's just make me a bit confused.
The lizards, they belong to the same species. They are just strategies, male strategies. And they are transmitted. I don't remember exactly what is the kind of the genetics that is behind that. But that is main strategy within the same species. So the mate, in fact, there can be daughters of male.
So you think
so it is within the same species. It's not really population dynamics of different species.
Yeah, but in principle, you cannot say that the population dynamics must be between different species.
Yeah, but what I'm...
No, I see,
okay.
I see. But
yeah. I really don't know what is the, say, the size of effect. But in fact, there is some, say, some genetic flow with the, say, across these different strategies.
Can I have another question? Nothing to do with that. If your second transparency, it was the exponential growth of the bacteria, and then that stabilization, and then the death. Why did they die? You remember that there was
this is simply you are in one beaker and there is no further nutrients. So when they end up the nutrient, they start dying. What is quite interesting is that the death is not really exponential. It's a sort of, say, faster than exponential, say. And that has not been studied that much, in fact.
There's another way of doing bacterial growth experiments. What is it called? Where you basically have a continuous influx of nutrient, and chemostat is the word. Yes, this was invented by Leo Szilard, actually, right, as a way of studying evolution and adaptation. So, has anybody done a long-term evolution experiment? Right, because you don't have to worry about weekends. You don't have to have a graduate student continually going pipette, pipette, pipette. You just have a continuous inflow of nutrients, and in the chamber there's a fixed population, because if they grow too much they just get swept out, etc., etc.
Yeah, but the problem is that, as far as I know, is that you have to be sure that the machine doesn't break down.
There is that, of course. But nobody's done a long-term evolution experiment there. Okay. Can I ask you to go back to the prisoner's dilemma game as a model for evolution of cooperation? Could you just give the context once again? Why is this a good model for the evolution of cooperation? Which, in what sense is it a model for?
In what sense? Okay, so
I always have trouble with this game theory approach, I must say.
Yeah.
So the idea is that if I think that it was due to the meeting of two minds. One is Hamilton and the other one is
The microphone.
Can use it. Yeah. So I think that the reason why there has been so much work on the prisoner's dilemma is due to the meeting of Hamilton and Axelrod. And the reason... Axelrod. Yeah, and the reason is that, so the idea is that if you look at prisoner dilemma, you assume that encounters are random and without future. Then this fact that the rational behavior is the one in which the defector wins, say, makes it impossible to maintain cooperation in a population. And so this was stated as sort of paradox by several people, and Axelrod introduced the idea of a repeated prisoner's dilemma.
In this repeated prisoner's dilemma, is one allowed to change the parameters?
No, just the fact that you know that you're meeting over and over again your neighbor, and if you are nasty to your neighbor, you are going to have a very hell of a life. So that's
very basically what happens. If you have a repeated prisoner's dilemma, it pays to be nice.
And then they made a sort of very funny, for instance, how to say, competitions, and they found that the competition winner was a tit-for-tat strategy. The idea, so in this case, you start changing the strategies depending on your history. And tit-for-tat is a strategy in which you start by cooperating, and then you retaliate whenever the guy you meet is not cooperating. And it turns out that this, in first encounter, say random encounter in a population, then you look who wins the most from a point of view of fitness is this tit for tat, because they cooperate if they meet, and if they cooperate, they increase their. So, but again, I'm not terribly convinced, even in the fact that it is possible to model cooperation among animals in this way. But it's again, it's a simple way in which you can introduce this frequency-dependent disruptive selection.
Yeah, we have to check the microphone. Yeah, but I think one of the important things is that the dilemma is in the fact that you are compelled to defect all the time. But if you don't, you are better off. But how can that happen if both of them are compelled to defect? So if by chance the two of them decide to cooperate, they get a better payoff, and then that can propagate. So there is. So you're compelled to defect and to be worse than you could be if you cooperated. So that's the dilemma.
But I think that it's much more realistic to look at what happens if you're playing the field. So that you have, and there you have also experiments, and again, I leave it to Paul to discuss about the meaning of these things. So the idea is that if you have a colony, for instance, colonies that need some enzyme in the environment in order to be able to absorb nutrients, then of course the individuals have a cost for producing this enzyme, but there can be free riders that simply exploit the fact that there are somebody else that is producing that. And in that case, you find that there are some subtle strategies that have been developed for the colonies to control the free riders in some way. For instance, getting the enzyme stick to the membrane and just be released very slowly in such a way that you know that the enzyme will be close to the source. That sort of stuff is probably more relevant. In this case, it's not just an encounter, but it's really playing the field.
Okay, yeah, the last question here, then we break for lunch.
So Luca, maybe just in words, could you remind us of what sort of revisit do you suggest for the fundamental theorem? You know, that term?
Well, say, again, the theorem has been considered as a paradigm for evolution, as a way of saying, well, evolution is an optimization. I don't believe evolution is optimization in any sense. So this is why I mentioned the fact that there were some disagreement on the relevance. One of the reasons is that you can only have evolution in very, very restrictive situations. Even if you consider, for instance, mutations, if mutations are relevant, then you find that only in very special situations you have a Lyapunov function. You have something that increases. It is so. Okay, so this is why I don't believe that much. But okay, that was.
Okay, So we should stop now and just one announcement. Food and drink, with one exception for the speaker, is not allowed in the auditorium. Okay? The water is okay, but not food or anything except for water, except for the speaker.
All right.
Let me your glass. We're very friends.